Hi everyone. So unlike animals in which growth happens due to the repeated division of cells throughout the body, cell division in vascular plants is confined to discrete but persistent populations of totipotent cells called meristems. Totipotent cells are those that have the potential to develop into any cell type in the organism. Growth continues throughout the life of the plant, making plants unlike many mammals such as humans, where growth ceases at maturity. For example, a plant such as a bristlepine cone can be thousands of years old, but still continue to send out new branches, leaves, and cones every year. Animals can respond to the world around them with their movement and behavior, but plants must respond to their world more slowly and can do so by changing and modifying their size and shape through growth. The shoot systems of plants are constructed in the same way despite their different appearances among the different groups of plants. Plant shoots are formed from repeating units or modules. Within each unit is a node and an internode. Nodes are where leaves are attached to the stem. The internode is the area between two nodes. At the very tip of each branch, commonly hidden beneath young leaves called leaf primordia, lies a tiny dome of cells called the shoot apical meristem. Here's the shoot apical meristem, and here are the leaf primordia. The shoot apical meristem, or SAM, gives rise to new tissues. In addition to the tissues of the stem, the SAM initiates leaves, thus the leaf primordia, and produces new meristems that will give rise to branches. Though the shoot meristem is a region undergoing active cell division, it stays a constant size. Only the cells at the very tip are able to divide. As the stem grows, the cells that have been produced and laid down via division are no longer in the vicinity of the shoot tip and they stop dividing. The length of the plant where cell division occurs is called the zone of cell division. Cells near the shoot tip maintain their totipotency, which is the ability to develop into any cell type, by the expression of meristem identity genes. These genes are expressed in response to short-range chemical signals produced by the cells at the very tip of the stem. Therefore, the meristem identity genes are only expressed in these cells near the tip, and that's what maintains their totipotency. In plants, most of the increase in cell size occurs after mitotic cell division is complete. Cells that are too far from the shoot tip to express meristem identity genes no longer divide, but they continue to grow. This results in a zone of cell elongation located just beneath the shoot apical meristem. The majority of stem elongation will occur in the internodes immediately below the shoot apical meristem. In the zone of elongation, the cells will grow more in length than in width, which is due to the orientation of the cellulose molecules in the cell wall. The majority of the increase in cell size and volume in the zone of elongation is taken up by the large central vacuole that forms in cells within this region. Most of the increase in cell volume is due to the uptake of water and solutes that will then fill the vacuole. Before vascular plants evolved roots or even leaves, they evolved the ability to form branches. Branching enabled early plants to produce more sporangia. In lycophytes and in ferns and horsetails, branching occurs when the shoot apical meristem divides in two. This gives rise to two stems, each with their own apical meristem. In seed plants, on the other hand, branches grow from axillary buds. Axillary buds are meristems that form at the base of each leaf. Until they receive signals to begin growth, the axillary buds will remain dormant and stay attached to the stem even if the leaves are shed. Before the evolution of leaves, early vascular plants were simple branching stems, and photosynthesis took place along the length of the shoot. In some of the early vascular plants, the apical meristems were lost on some side branches, and they became flattened into a single plane. New meristems then developed that permitted photosynthetic tissue to form between the side branches, giving rise to the development of leaves. The photo on the right shows Archaeopteris, which is one of the first plants in the fossil record to show flattened leaves. Typically, it is the function of photosynthesis that comes to mind when we consider leaf function. However, leaves can also be specialized to perform other tasks. Bud scales, seen here, are specialized leaves that provide protection to the shoot apical meristem from desiccation and damage for plants that overwinter. Tendrils, like you can find on pea plants, are modified leaves that allow plants to climb or attach to structures. The trap on the Venus flytrap plant is formed from modified leaves, and they catch insects to provide the plant with a source of nitrogen compounds. Here's an example of colored leaves. In this bougainvillea, the brightly colored leaves surround the flowers and enhance the ability of the plant to attract pollinators. Like leaves and branches, flowers form from meristems. Flowers can be produced at the tip of a plant or shoot as a result of the conversion of the apical meristem to a floral meristem, or as the products of axillary buds. So here on the left is an apical meristem that has been converted into a floral meristem. This conversion, or flowering, is triggered by phloragen. This is a protein that is produced in the leaves and then transported via the phloem to apical meristems and axillary buds. Phloragen then triggers the transition from a shoot meristem to a floral meristem by turning off meristem identity genes and turning on genes that are responsible for floral identity. There are 3 classes of such genes. They are called A, B, and C. These genes are expressed in overlapping rings around the meristem. You can see here a diagram of their expression. These genes serve as master controllers for the development of the floral whorls. The identity of each whorl is controlled by the expression of a specific combination of genes. So for example, in cells that express only C genes, these will develop into carpels. But if the cells express both C and B genes, they will initiate the development of stamens. This is known as the ABC model of flower development. With the move onto land, plants had to evolve mechanisms to control the timing of reproduction and the development of flowers in environments with distinct seasons. These timing mechanisms help to ensure that plants are both large enough to support the production of seeds and that they have enough time to do so before winter starts. In the early 1900s, scientists from the U.S. Department of Agriculture determined that plants were able to determine day length. They coined the term photoperiodism to describe the effect of day length, or the photoperiod, on flowering. Short day plants flower only when the day length is less than a critical value. When the light period exceeds this threshold, a short day plant continues to produce new leaves, but no flowers. For example, this morning glory, we have flowers with the shorter day, but only vegetative growth with the long day. Conversely, long day plants will flower only when the day exceeds a certain length. A frequently used example used for long day plants is the common model organism Arabidopsis. Not all plants are photoperiodic, however. Flowering in day-neutral plants is independent of any change in day length. Instead, they may initiate flowering when they reach a certain overall developmental stage or age, or in response to environmental stimulus other than light. Plants are able to measure day length by the use of photoreceptors and the plant's circadian clock. Photoreceptors are molecules whose chemical properties are altered as they absorb light. One of the photoreceptors that plays a role in photoperiodism is the phytochrome, which we have come across before with seed germination. Circadian clocks are oscillating biochemical mechanisms found in all organisms that coordinate with the day-night cycle. The absorption of light by a photoreceptor produces a signal that triggers changes in the cell's metabolism and alters patterns of gene expression. We can see the pattern of seed germination and flowering with short and long day plants if we zoom out to look at a year. The blue line on these graphs represent the length of day, with the dotted line denoting the critical day length cutoff. Short day plants can germinate in the spring, but not typically flower until late summer or fall. Short day plants do not flower in late spring or early summer, despite the day length being below the critical cutoff, because the plants at this time are too young or they're too small to respond to day length and be able to support seed production. Long day plants will typically flower in late spring or early summer, around the summer solstice, as the day lengths are the longest around this period. There are some long day plants that also require a period of cold weather in order to flower. This allows these plants to germinate one year, grow throughout the summer, but then not flower until early summer the following year. Plants that undergo this timing strategy can grow larger before undergoing reproduction, which can increase their chances for successful pollination and support for seed production. Chemical signals such as hormones influence both the growth and the development of plant cells. Growth is an irreversible increase in size. Development, on the other hand, includes the formation of new tissues and organs. Development also includes the differentiation of cells. There are 5 major hormones recognized in plant growth and development. They are auxin, gibberellic acid, cytokinins, ethylene, and abscisic acid. They set up and maintain the basic patterns of plant development, as well as coordinate the growth and development of plants in response to internal and external environmental stimuli. Hormones can be synthesized throughout a plant, and they can affect the cells in which they are produced, as well as cells located in distant regions of a plant. Hormones influence the growth and differentiation by altering patterns of gene expression, as well as rates of cell division and cell expansion. The effect of one hormone is often affected by the presence of one or more other hormones. Additionally, how a cell will respond to a specific hormone is influenced by its position and developmental stage. This combinatorial effect is why a small number of hormones are able to shape the growth and development of plants. We will focus on a few of these hormones in this lecture and in the next, but make sure to take a look at this summary table to see where these hormones are synthesized and found, and what effects they have on plant physiology and development. Auxin was first identified by its ability to cause shoots to elongate. Later, it was also identified as the hormone transported preferentially from young leaves to the differentiated vascular tissues in the stem. The movement of auxin creates a template that guides vascular differentiation. This is due to the fact that the movement of auxin can be controlled. So how do plants control the direction of auxin movement? So auxin is secreted by cells in young, expanding leaves. And while it is in the cell wall, it doesn't carry a charge, and so it can easily enter surrounding cells by diffusing directly across the plasma membrane. However, the pH of the cell's cytoplasm is higher than the pH of the cell wall. This causes the auxin to lose a proton or hydrogen ion. The negative charge that then results prevents auxin from moving easily back across the plasma membrane. In order for auxin to leave the cell, a plasma membrane transport protein known as a PIN or PIN protein is then required. By restricting the placement of PIN proteins to only one face of the cell, plants can control the direction of auxin movement. PIN proteins are located only on the basal side of each cell. This is the side furthest away from the shoot tip. These proteins are here in yellow in this diagram. This means that auxin can only leave the cell at the basal end. This creates a polar gradient with auxin only traveling in this direction. Auxin stimulates cells to elongate and increase their production of PIN proteins. Auxin transport results in linear rows of elongated cells, since outflow from the basal side of one cell leads to enhanced auxin concentration in the cell immediately below. The elongated cells that carry auxin from the developing leaves to the stem differentiate to become procambial cells. Procambial cells then give rise to the xylem and phloem of the vascular tissue. Here in this diagram to the left, you can see the strips of procambial cells in purple running down the leaf primordia. In the younger leaves, the procambial cells are still differentiating, and the connection to the stem is not complete. However, in this older leaf, the procambial cells are connected to the vascular tissues of the stem. As auxin moves by polar transport out of developing leaves, it is drawn towards the vascular tissues in the stem by diffusion down its concentration gradient. The vascular tissues in the stem are a region of low auxin concentration because they transport auxin towards the roots faster than it can move through the undifferentiated cells to the vascular tissues by diffusion. The polar transport of auxin out of the cell and diffusion between cells is responsible for establishing the vascular connections with the stem that allow leaves to grow and function. The vascular system of an elongating stem is built from the successive addition of procambial strands that initiate in the developing leaves. The simplest arrangement of vascular tissue, seen in lycophytes, is a single cylinder of vascular tissues with xylem at the center and phloem on the outside. The procambial strands that connect each leaf to the stem merge with this central cylinder. In seed plants, and that's what we have here in this diagram, they exhibit a few different configurations. One of the most common is a ring of vascular bundles near the outside of the stem. The bundles have the xylem conduits towards the center of the stem and phloem towards the outside of the stem. This setup, with the stiff xylem tissue close to the exterior of the stem, contributes to the support of the plant body and helps to prevent the plant from bending over. The region between the epidermis and the vascular bundles is made up of ground tissue and is called the cortex. And the region inside the ring of vascular bundles is the pith. The pith is also made from ground tissue. Branching allows plants to support more leaves, but if new branches are too close together, their leaves can overlap and shade out one another. Axillary buds are found at each node, but only a few will ever develop into branches. The interplay of hormones communicates to axillary buds when conditions are right for the growth of a new branch. Removal of the shoot tip stimulates the outgrowth of axillary buds along the branch or stem, suggesting that the shoot meristem suppresses the growth of axillary buds. This suppression is referred to as apical dominance. Studies have found that if the shoot tip is cut off and a paste containing auxin is applied to the cut end of the stem, the axillary buds will remain dormant. On the other hand, if a substance that inhibits auxin transport is applied to the shoot tip, branching is induced from the axillary buds. Branching is therefore suppressed by the movement of auxin from the shoot tip towards the roots. Apical dominance prevents branches from being too close to the shoot tip and also allows new branches to form should the shoot meristem be damaged. The development of axillary buds is affected by multiple hormones. Auxin is able to suppress the growth of axillary buds by inhibiting the synthesis of cytokinins. Cytokinins are hormones that stimulate the outgrowth of axillary buds by increasing the rate of cell division. When the apical meristem is removed, cytokinin synthesis increases in the stem nodes, triggering the growth of axillary buds. Recent studies also suggest that control of this process is complemented by another hormone, strigolactone. If you have any questions, please let me know via the Q&A discussion board.